Can you think of some problems that cells need to consider when replicating their DNA? Cells need to make sure you get exactly 2 copies which are identical. They have to make sure replication happens only once per cell cycle, which is during S phase, and that everything happens quickly enough that rapidly dividing cells won't be held up while they wait for DNA to replicate. After all, you've got about 3 billion base pairs in the cell. Cells also have to minimize mistakes, such as single nucleotide changes or expansion of repeated sequences, and they have to fix mistakes that occur. And lastly, they need to deal with the issue of not losing DNA at the ends of the chromosomes, which I'll get back to in a minute. Alright, so where do we start? Well, DNA replication starts at an area called the origin of replication. How many of these are there? Well, there's only one in a prokaryotic chromosome, but eukaryotes have a bunch of them since they have a lot more DNA, and it would take forever with only one. At this origin of replication, the 2 strands of DNA separate and forms what looks like a bubble. Each end of this bubble is the replication fork, and as replication progresses, this will expand outward as the new strands of DNA are formed. Let's take a look at one of these replication forks. So the blue is the parent DNA, and I'll show the newly synthesized DNA in red. The 2 strands replicate in slightly different ways. One replicates continuously, called the leading strand, and the other replicates in pieces, called the lagging strand. Now why can't the lagging strand create its new daughter strand continuously? Well, this has to do with the polarity of DNA. Do you know which end of each strand here is the 5' and 3' end? You might want to pause the video and think about it for a second. So DNA is always synthesized in the 5' to 3' direction, and that's referring to the direction of the strand being synthesized, not the original. On the leading strand, this new piece of DNA can be synthesized continuously, since the replication fork that it's going towards is at the 3' end of the strand being synthesized, and therefore the origin of replication is at the 5' end. However, the lagging strand has to be synthesized in the opposite direction of the replication fork is moving. So as the replication fork progresses, it has to keep starting over. Do you know what each piece of DNA in the lagging strand is called? That would be an Okazaki fragment. Okay, let's look at some of the enzymes involved. I'll mostly go in chronological order. The first is helicase. So what does helicase do? It unzips the DNA and allows the bubble to expand. It's actually a little known fact that helicase is the most fun of all the enzymes since it spends all day unzipping genes. Get it? Sorry, bad joke. Anyway, helicase does this just by disrupting hydrogen bonds between the 2 strands. It doesn't make or break any covalent bonds. Since helicase is moving along with the replication fork, something is needed to prevent the strands from reannealing to each other, which complementary strands of DNA like to do. Do you know what protein does this? This is where a protein called single-stranded binding proteins comes into play, which is just that, a protein that recognizes and binds to single-stranded DNA to prevent it from binding to its complementary sequence in the other strand. The last proteins I'll mention on the subject of opening DNA up are the topoisomerases. These are proteins that regulate supercoiling of DNA. An analogy you can use to think about this is think of 2 strands that are anchored at one end, but are then wrapped around each other a few times. If you pull the 2 ends apart, they'll be wrapped tighter and tighter until pretty soon you can't pull them apart at all anymore. This is why you need topoisomerases. Now, regulating supercoils is a vital process in cells, so if you inhibit them in bacteria using fluoroquinolones, the bacteria are unable to divide. Alright, back to the synthesis of DNA. So what enzyme is in charge of synthesizing the leading strand? That would be DNA polymerase III, which synthesizes the leading strand continuously towards the replication fork. Once again, always 5' to 3'. Okay, so one more thing that needs to be done before we can make the lagging strand is adding RNA primers. There are no DNA polymerases that can initiate DNA synthesis from scratch. They have to build off of existing DNA or RNA. So what enzyme can help us here? That would be primase, which can synthesize RNA from scratch, by which I mean that it can add ribonucleotides to their complementary DNA bases and connect them together. Once a primer has been laid down, our friend DNA polymerase III can jump in and start adding deoxyribonucleotides to it, just like it did for the leading strand. DNA can only be synthesized in a 5' to 3' direction. So DNA polymerase III starts from the RNA primer and builds towards the 3' end of the new strand. So as DNA polymerase is making each new strand, it proofreads its work, and if it realizes it made a mistake by adding the wrong nucleotide, it can remove it by using its 3' to 5' exonuclease activity. This is of course the opposite direction of DNA synthesis, since it's going backwards to remove a mistake. After the lagging strand has become a continuous stretch of alternating RNA primers and Okazaki fragments, the only thing left is for a different polymerase, which is DNA polymerase type 1, to remove the RNA primers using its 5' to 3' exonuclease activity, and fill in the gap with DNA as it goes. Polymerase 1 has the same synthesis and exonuclease functionality and polarity as polymerase 3, but has the added ability to remove RNA primers. Alright, so one last enzyme here. DNA ligase will seal the hole between the DNA fragments. Keep in mind, this is between the fragments of the same strand of DNA, not connecting one strand to its complementary strand in a double helix. The last problem we have is what to do about the ends of the chromosomes. The leading strand is no problem since it can just synthesize DNA all the way to the end, but the lagging strand is dependent on RNA primers, and the last RNA primer can't be filled in with DNA once it's been removed, since DNA synthesis would require another primer to build off of. Therefore, a few hundred nucleotides of DNA are lost each time the cell divides. The solution is to use telomerase, which is a ribonucleotide protein that repetitively adds a 6-nucleotide sequence, TTAGGG, to the end of each lagging strand, creating non-coding junk DNA that can be lost without harming the function of the cell. In adults, telomerase is only expressed in cells that need to divide a lot, such as stem cells and some cells in the immune system. Although mutations that can cause telomerase to be expressed in other cell types are one of the things that are necessary for neoplastic transformation, since without telomerase, even cancer cells will lose the ability to divide after a while.